All right. Good morning, everybody. Thank you for joining us for the second day of this year's ISB Microbiome course. My name is Alex. I'm a senior graduate student in the Gibbons Lab, and I'll be the instructor for today's portion of the course, the focus of which is predicting personalized pathogen engraftment with metabolic models. Before we get started, let's make sure you're set up with the course notes. You can open these slides using the URL shown here. Should also be in the email that was sent to you prior to the course. The TAs have also posted the link on the Slack channel where you can find it as well. As a reminder, similar to yesterday, this course will be taught across 3 modules. The presentation slides we're currently looking at will contain explanations of the logic and methods used in the course, as well as links to pertinent materials related to the course. The analysis and modeling, as well as visualization of the results, will take place in the course notebook, hosted in Google Colab. You can navigate to the notebook using the link in the next slide, or from the email sent to you before the course. Finally, the Slack chat is available to you at any time during the course for questions or comments. So if you have any questions, don't hesitate to post them there, and the TAs will be happy to help you out. For the next part of the course, we'll be revisiting the theme for this year's course, predicting personalized pathogen engraftment with metabolic models. Clostridioides difficile, or C. diff, is a leading cause of morbidity and mortality globally. In the US alone, CDI affects nearly 500,000 individuals annually, around 6% of which are fatal. As you can see in this graphic, CDI disproportionately affects the US, Canada, Australia, parts of Europe, as well as parts of Africa and other regions of the globe. But while CDI is a serious issue, C. diff isn't always a bad guy in every context. In adults, estimates suggest around 4 to 15% may be asymptomatic carriers of C. diff, while in infants, the rate of colonization is much higher at around 30%. Infection develops following disruptions to the microbiome, such as antibiotic use or the onset of diarrhea. In these contexts, C. diff spores allow it to persist, allowing it to persist following the disruption. And without competition, C. diff can bloom, which ultimately leads to CDI. Now for today, we would like to be able to predict C. diff engraftment using Microbiome, a metabolic modeling tool developed here in the Gibbons Lab. To do so, we'll be using the following workflow. As a reminder, we'll be working with samples from healthy fecal microbiome transplant donors, as well as individuals who had CDI and received an FMT. To do this prediction, we'll first import the abundance tables that were generated in yesterday's tutorial and introduce 10% C. diff to all of them as a challenge. This will act as a sort of in silico invasion assay to test the susceptibility of our samples to infection, or in this case, engraftment. We'll then estimate growth rates and fluxes using a method that we'll explain today in the class. And then we will use this information to assess the susceptibility of our samples for engraftment. We'll also look at the flux predictions to try to understand what niches C. diff might be occupying it when it does successfully colonize. And with that, before we jump into more of the course material, let's go ahead and take a look at the notebook and get things going there. All right, so you should be able to see the notebook. Now, the first thing you'll want to do is go to File and save a copy of this notebook in your drive so that you can edit it. I'll go ahead and close the original one, and then I will rename this one Alex's Microbiome 2023. Great. So if you attended the course yesterday, you'll be already familiar with the structure of this notebook. In case you did not, let's go ahead and review that briefly. So in this notebook, you will see there's text that's sort of explaining what's going on. And then there's these little gray portions, which are code cells that you can click this little play button to run. Now, before we actually, let's go ahead and get this first one going. Let's see, first I have to connect. Once it's connected, you can see you get this little check mark. Then I can go ahead and run this first code cell by clicking the play button. This will go ahead and download the course materials from the GitHub, and then it will move us into that directory. Now, some other important details here. If you look at the side of the notebook, you can see this first little object, the table of contents. You can click it. And this just sort of gives you a nice index to kind of jump around, so you can kind of see what the different portions of the notebook are, and you can, like I said, jump around to those. And then also importantly, we have this little folder here. This is our directory. You can see it looks like the materials folder was successfully downloaded, and now you can see everything that's there. So now that we have the materials, we want to install Microbiome, which is the tool we'll be using today, and also allow interactions with Chime, which is included in this this download. So you can see this is running pretty quickly. Once this is done, we're actually going to go ahead and move through the process of importing the data and getting the models building. This is kind of a slow process, so it's good for us to get this going now, and then we'll jump back into the course slides and explain what's happening. But just as a little preview here, this graphic is kind of giving you a flavor for what we're going to be doing today. Just to touch on it briefly, we're going to be first importing the abundance tables that were generated yesterday. If you weren't participating in the course yesterday, that's okay. We're actually going to be pulling these tables from the treasure chest. We're also going to be using a set of curated metabolic models to map to our abundance table. And then we're going to use a Microbiome function, Microbiome build, to build our metabolic models. That'll be the first step. And then once that's done, we'll come back later, import a medium that is representing the diet for our samples. And then we'll use this grow function to estimate the growth rates and fluxes. So first things first, let's kind of orient ourselves with Microbiome. So here we're just importing some test data to give you a flavor for how these tables are going to look. So here we have an example table. The first column is the ID. It's just a unique identifier that sort of gives you a name for the taxa that are being represented in the models. Then we have some other identifiers for those associated with those taxa, like the genus or the species. There's some information regarding the number of reactions and metabolites in these models, as well as the sample ID. In this case, you can see we have two samples, sample 1, sample 2. The first one has 4 different strains of the E. coli, and then the second only has 1. And then you can see here the abundance is telling us a little bit about the who's there and to what degree. Great. So now let's go ahead and import our table from yesterday. To do that, we'll run this next little code block. You can see that's pretty fast. And so this table is representing the samples from the 8 individuals that we worked with yesterday. These are broken across 2 states, individuals that had a CDI and received an FMT, and individuals who were FMT donors. That from this table is not very clear though. So next we'll want to import our metadata and merge that with the taxonomy table using this little code block. And you can see now we have all the same information as before, but now we have a bunch of additional information, including the timestamp when the sample was collected, how many days relative to FMT the sample is, as well as the disease state of the individuals. Great, so now that we have our table, next as I mentioned before, we're going to want to challenge our samples by invading them with C. diff. To do this, we're going to be introducing 10% C. diff to the samples. This is a little bit of a more complicated coding block, but I'll try to walk you through it. So here we've defined a function called invade, which is what we're using to invade our samples. So to define the function, you'll want to just click this play button. It's not actually going to do anything in terms of showing you a result, but it will let us use the function now. So for this function invade, first we're going to want to give it the data that we're working with. That's the table that you just seen. We're going to want to specify the invader. In this case, it would be Clostridioides. And then we'll also want to define the relative abundance of the invader. And so what's going to happen here is first we're going to define, first of all, this is just some text that sort of explains the parameters. So first we're going to want to define our output. It's a data table, an empty data table called invaded. Next, we're going to want to loop through the data table one sample at a time using this pandas group by function. This will, like I said, kind of chunk out the data into chunks for where they all have the same sample ID. Next, we're filtering out low abundance genera. Normally, this threshold that we're using here, 5%, would be much lower, but to make the process run at within a reasonable time frame for the course, we've chosen 5%. Then here you can see we're filtering out Clostridioides. That's because for the purpose of invading with Clostridioides, we'd actually want some of the samples, as you might have seen yesterday, have it already there. So because we want to introduce at a fixed percent, we want to first remove it. And actually, to be consistent with the function here, this should actually be invader. But you can leave yours as is if you want. It won't make a difference for this purpose. Next, we're going to want to calculate the abundance that the invader should be at to achieve the desired relative abundance. Then we're going to gather some information for the sample that's going to be relevant, and then introduce the invader specific information. And then we're going to add it to the end of our data table and recalculate the relative abundance of our samples to account for the invader. And then finally, we're going to save that in our output invaded and return that. I'm going to run this again since I updated the little code snippet there. And now we're going to run it here by introducing Clostridioides at 10%. And then this second line here is just filtering the data table to show us only the rows where Clostridioides, where the genus is equal to Clostridioides. And so what you can see is that across all 8 of our samples, it successfully introduced Clostridioides, and the relative abundance is 10%. Great. So now that we have this table constructed, we could go ahead and start building our models. That's going to happen here in this next code block. And so an important thing to note here is we're feeding it the table invaded that we just generated. We're also giving it, we're telling it what the database is that we're going to be using. In this case, the Agora1 database, which is a curated database of metabolic models. We're going to be creating a folder called models, and then we're telling it which solver to use and the cutoff and how many processes it should use. One important thing to note about the solver, this actually, because it's solving a very complex problem, I'm going to get this running. The choice of solver is very important. In this particular case, because we're running on Google Colab, we're using a publicly available solver, which isn't quite ideal for this problem, and is part of the reason why we've tried to keep this cutoff high so that things will run in an appropriate time frame. If you're running this on your own server, you might want to invest in a more powerful solver. So you can see this is running now. This is going to take about 10 minutes to go. So while this is going, I'm going to hop back over to the slides. Great. And now we'll kind of run through some of the background on what we were just doing. So as I mentioned, yesterday we used QIIME2 to analyze sequencing data, starting from raw sequences and ultimately generating a table of amplicon sequence variants, or ASVs. That was the table that we imported and used to start building our models. As you've seen in the table, we assigned taxonomy to our ASVs. Also, diversity metrics were used to compare samples. Today, we'll look at functional analyses of the gut microbiome. That is, rather than looking at what populates the microbiome, we'll be determining what is happening in the microbiome, which is not always fully explained by taxonomy alone. When we first say functional analysis, a few things might jump into mind, like, for instance, some of the omics methods listed on the slide. Most commonly, to determine the function of the microbiome, one might do a metagenomics study, which in this case is not what we did. We have 16S data. But if you did have metagenomics data, what you might be able to get from that is to see what genes are present in the community. Mapping these genes to a reference database is fairly simple, and from there we might be able to infer what reactions could take place in the system and put together a network of reactions taking place. This would be great, but there's a caveat in that the gene abundance data doesn't always tell you the full story. That's because gene abundance really only reports the metabolic capacity of the community. If you imagine a microbial community with really high abundance of starch degradation genes, for example, you might infer that a lot of starch degradation pathways are active in the host. However, if the host isn't actually eating any starch, then clearly this inference is wrong. Thus, gene abundances tell you what could happen, but they don't necessarily tell you what is happening. Thus, gene abundances are cool, but it's not necessarily what you really care about. And they don't necessarily tell you the whole story. So today we'll explore an alternative approach to analyzing the function of the microbiome, that is, predicting metabolic flux. Fluxes will tell us more about the actual metabolic behavior of the microbiome than abundances can. Great. So what are fluxes? Well, rather than abundances, which are a measure of concentration, fluxes are a measure of mass conversion into the system and through internal reactions. Since they represent a rate rather than a concentration, fluxes are measured in units of concentration per unit time. For instance, millimole per hour. In bacterial communities, we typically scale flux by relative abundance to account for differential metabolic contributions by more or less abundant taxa. So why am I claiming that fluxes are more informative than abundances? Well, if we look at the example on the right-hand side of the slide, what we see is that glucose is being imported into the cell and then being fed into glycolysis and then eventually the TCA cycle. We see that the cell in question is importing a ton of glucose, but that glucose is almost immediately being converted into pyruvate. If we were to measure internal glucose levels, they might actually be quite low. This would lead us to incorrectly assume that glucose isn't required for the cell to grow or just isn't that important. Obviously, that doesn't tell the whole story, since glucose is clearly being utilized, as you can see from the flow in this graphic. What would be more informative would be to see the flux of glucose in the cell and the resulting pathways that are activated and metabolites produced. Clearly, we can learn a lot from looking at fluxes in the system like this. The problem with measuring fluxes, however, is that it's laborious and can be quite costly. Longitudinal metabolomics, for instance, can provide a way of measuring concentrations over time and allow us to calculate fluxes. But this can be a pretty big effort and fairly expensive. Furthermore, it's often limited to those metabolites that are included in your metabolomics panel, and often requires that you're able to grow your organism or community of interest in a defined medium. Another alternative here is the use of isotopic labels, wherein you feed the community or an isolate isotopically labeled substrate, for instance, the glucose, using natural isotopes of carbon or nitrogen, and then you look for the appearance of those isotopes in other cellular metabolites. That's great, and can be actually a very informative approach. However, that can also be even more costly than the standard metabolomics analysis, and is still limited to just those metabolites being investigated. At most, you might be able to do 1 or 2 isotopically labeled substrates. So what we need is a way to easily and accurately estimate fluxes without the labor and expense associated with these methods. Luckily, there's already a computational tool for that. To do that, we are going to be using a method called flux balance analysis, which has become fairly popular in recent years in microbiome research. This is a computational method that uses genomic information to infer the most likely set of fluxes in a biological system. We can use this to predict the metabolic behavior of a microbe using genome-scale metabolic models. All right, so let's take a closer look at FBA and how it works, and how we can use it to infer the fluxes that we're interested in. FBA is a powerful computational tool. It allows us to infer fluxes through reactions in a system by reducing the possible solution space of fluxes to a biologically relevant one. FBA makes the critical assumption that the system being modeled is in steady state. That is a state of constant flow, which just means that the inflow and outflow of reactants in the system are equal. A good analogy here is maybe like opening a window in your house. You'll have air flowing in and air flowing out, but you won't suddenly have more air flowing in than out. If you did, then your house would expand and eventually pop like a balloon due to the buildup of pressure. Obviously, that doesn't happen. Thus, in that particular example, the steady state assumption holds. And in general, this assumption in the context we're interested in kind of mimics this scenario. Now, if we look at our example reaction here, the formation of water from hydrogen and oxygen, what we can say in the steady state is that stoichiometrically, the amount of hydrogen and oxygen consumed are equal to the amount of water produced. We can then represent this mathematically with the equation below the reaction. Reactants with negative coefficients and products with positive coefficients, balancing to 0. Repeating this for all reactions in our system and factoring out the fluxes, we could end up with a stoichiometric matrix designated S, as shown on the right, that represents all the reactions, as well as an unknown vector of fluxes designated V, which we would be solving for. To satisfy our assumption of constant flow, we would then set the dot product of S and V, which is just a form of matrix multiplication, equal to 0. That sets up our system of equations. Additionally, if we want to constrain our solution space, we can put bounds on the fluxes using information, say, for instance, the reversibility of a reaction, which might limit the value of the individual fluxes from, say, 0 to some upper bound. That would actually limit our solution space and kind of give us what is thought of as a flux cone, which is the space in the multidimensional flux space that includes all sets of fluxes satisfying our constraints. You might notice, however, that there's an infinite number of solutions in our flux cone that could satisfy these constraints. So what's next? Well, the goal of FBA is to reduce the flux space to one that is biologically relevant, so that it mimics the actual behavior of the system as well as possible. But how do we find the optimal solution within the possible solution space? To achieve that, a first step is to look at growth rates. We know that bacteria can only be present in the system if they grow. Otherwise, they would be excreted and no longer exist in the system. In practice, this means we can say we want the solution in the flux cone that corresponds with maximum biomass, since this is generally the biological goal of growing microbes. Biomass production can also be represented as a flux, since it is an essential it is in essence a measure of mass conversion. What's great about flux balance analysis is that we can add information from a number of sources to constrain the problem and determine our optimal solution. As we mentioned, we can use the reactions present in the system to build a stoichiometric matrix, mathematically representing the reactions taking place and how they interact with each other. That's depicted in the upper left here. Once we infer the stoichiometric matrix, we can well, let me take a step back. We can actually infer the stoichiometric matrix from the genome of a microbe by looking at the gene abundances to determine what reactions are potentially taking place. Then, as we've mentioned, we make the assumption that the system is at a steady state. In practice, this means the bacteria is in exponential growth, as the growth rate is constant and biomass production is constant. So we include this assumption to arrive at a possible solution. Additionally, reversibility of reactions can be determined from thermodynamics, as many reactions can only occur in one direction, thus limiting the space of possible fluxes. And then finally, we can add additional constraints to the imports of metabolites going through the system, which we can leverage to represent the metabolites available to the system for growth. In practice, we can use this to model the effects of different diets on the microbiome, thus adding relevance to the predictions being made by FBA. Cumulatively, these constraints help us find the region in the flux code that represents maximum biomass, as shown in red. The art of FBA is then to find a single solution in that orange or red space that is maximizing biomass, that is biologically relevant. There are several methods available to find a single solution, but today we'll focus on these last two in the list, as they are the ones leveraged by Microbiome. One such method is minimizing the imports into a system. Since the microbiome can be a highly competitive environment, this is biologically intuitive, since the most successful residents in the microbiome would not need to rely on importing lots of metabolites to survive, rather subsisting on a smaller subset. Another common approach is the use of parsimonious FBA, also known as PFBA, which basically minimizes the enzymes required to satisfy the constraints. Again, this is biologically intuitive, since bacteria are efficient beings and don't like to expend more energy than necessary on making enzymes. Really, the method you use is related to the question being asked and the system being modeled. Is it competitive? Is it resource limiting, etc.? I encourage you to look more into these methods if you're interested in conducting FBA on your own, as different methods might be more or less applicable to different situations. Great. So to this point, we focused on making genome scale metabolic models. To make metabolic models of the microbiome community, or metagenome scale metabolic models, we use a tool called Microbial, which extends flux balance analysis into microbial communities. To initially build the models, we need to pass in the relative abundance of the bacteria in the sample, as I mentioned earlier. Due to sequencing efficiency differences, there may be some bias towards some bacteria over others, but in general, we assume the abundance from sequencing data should be more or less representative of the community. Microbiome will then map this abundance data to a database containing hundreds of genome-scale metabolic models of common gut microbes. As I noted earlier, we're using the Agora database, which is a well-curated database for these models. Microbiome then uses the reconstructions of those taxa present in the sample to build a massive stoichiometric matrix, like the one we discussed earlier, that includes not only the internal reactions with each taxon, but also the exchanges between them and external reactions, which in this case is the host. We can then specify a diet that is representative of the food being eaten by the subject of the model, and use FBA with an additional regularization step to calculate unique growth rates for all the bacteria based on the process we've outlined previously. Following this, estimates of all the fluxes in the system can be generated based on those growth rates, returning a most likely flux distribution. This gives us a huge amount of output data, which we can then use to make detailed and interesting predictions about imports, exports, and the inner machinations of bacterial reaction networks, codependencies between bacteria, and a testing ground for potential interventions. I showed this to you earlier in the Google Colab, but here's an outline of the typical workflow for building metabolic models and simulating growth as well as flux. As I noted, first we will build the models using abundance data and the database of genome scale metabolic models. Then we apply a medium representing available metabolites. And finally, we simulate growth of models using a multi-step FBA method that we'll cover shortly. What's great about MyCom is that there are two interfaces in which you can use it, both in Python and QIIME 2. Today, we're going to be using the Python interface as it works pretty seamlessly with Colab, but you can also perform any task with either of the interfaces, either on the command line using the QIIME 2 interface or in a notebook as we're doing today. Either actions like building or growing models for producing visualizations can be done using these interfaces. And it's pretty easy to switch between the interfaces, between steps, as MyCom can readily read QIIME artifacts and vice versa. Great. So with that, I think our models will likely have its finished building. So let's take a peek. And yeah, looks like the model's finished building. That's great. So now we are going to want to import our medium. In this particular case, we're using a medium that represents a sort of average Western diet. More accurately, this is an average European diet. And so what this does is it takes the composition of the diet and it breaks it down into fluxes of various metabolites that will then be available to the community, thus constraining the solution space for the growth rate estimates. Next, we're going to feed the manifest that was generated above here. And actually, let's take a quick look at this manifest just to see what it looks like. Again, we have our sample ID and some of the metadata. And if we scroll over to the right, you can see the fraction of genera that were found, as well as the fraction represented in the relative abundance. You see in this particular case, we do a pretty good job for most of the samples. Again, we have used a cutoff that removed a lot of the very low abundance organisms, so this is slightly inflated. But in general, you can expect somewhere between 80 and 90% to be captured, which is usually a good indication that your samples can be modeled. You do see, though, we do have one somewhat problematic sample here. We found around 70% of the genera, but is only capturing about 50% of the total relative abundance. Sometimes you'll actually get an error if the found fraction is below 50%. In this case, it was just above that, so we didn't receive that error. But that's just something to keep in mind. If you get a strange result, it might be because we aren't accurately representing the sample. Great, so we have that. We also have this medium. Now we can grow the models. We're going to be using this function grow. It takes the manifest that was generated from the build step, and it's going to want to know where that folder of models is that got built. You can actually, if you look to the left, see that folder here. We take a peek inside. You can see the models were built for each one of the samples in a compressed format, this .pickle format. Next, we're going to want to specify this parameter called the trade-off, which I'll explain a bit more when we jump back into the slides. This will essentially is a really important feature for the simulation that's being done, and has a very strong impact on the results. So it's important to pick the one that is most meaningful. In this particular case, we already know from previous tests that 0.8 is a good value to use. So yeah, we'll go ahead and get this guy going. And then it's going to save the results in output called growth.zip. again, compressed format because the data or the results we're generating are fairly large. Again, this is going to take about 10 minutes. So let's hop back over to the slides. Great. So now that we've covered classic flux balance analysis used for genome scale metabolic models, and it seems pretty straightforward, right? You maximize the growth rate and get your answer. So can we just do that same thing for the community metabolic models? Well, not quite. In genome scale metabolic models, we have a single growth rate. But in community models, we have multiple growth rates, one for each taxon, as well as a community growth rate calculated by multiplying the relative abundance of each taxon with its growth rate and summing them all together. If we try to maximize the community growth rate, you'll find quickly that we run into problems. Oops. So yeah, with classical FBA, we actually find that for a single taxon, this approach works fairly well, and it's why it's gained such popularity. You can fairly accurately predict the growth rates of individual bacteria when you are growing them in well-defined media, and you have a good annotation of their reactions that are present. As an example of this, we can see results from a study that compared empirically measured fluxes with those predicted by FBA across a series of evolved E. coli strains. This evolution was done to try to maximize their efficiency of growth. And what we see is that the predictions agree quite well with the empirical estimates. And the evolved strains are able to achieve growth rates that are on the order of 90 to 95% of the theoretical maximum. And partly that's because E. coli is a very well-characterized organism. They're growing it in a defined medium. And so, this is sort of the ideal case. However, it's when we consider communities that things start to break down. Classical FBA doesn't do so great here. One of the issues is that given multiple taxa, optimal community biomass may not be achieved by, optimal community biomass can be achieved by various linear combinations of the growth rates of the individual taxa. That is, there's a non-unique solution space for the growth rates and fluxes, as kind of noted here in the lower left. So you might have a solution, for instance, where the optimal community biomass is achieved by only one organism growing, potentially the fastest grower. Additionally, experimental results have suggested that optimizing community biomass may be a slightly flawed approach in this particular context. As empirically, many communities achieve lower overall biomass as diversity increases due to competitive interactions and other factors, as is shown on the right. You can see, for instance, in this community of bacteria shown in the red, as you increase the diversity of the community, the total biomass achieved by the community actually is decreasing. And the same is true to an extent for a yeast-based community as well. But all's not lost, which is why we're here. This is where cooperative trade-off flux balance analysis comes in. This is a method that was developed alongside with Microbiome. This approach is able to generate unique, biologically relevant solutions to the community growth rate problem posed just a moment ago. Now let's try to understand how. First, let's consider the growth curves on the left. As many of us are probably familiar, bacteria and other organisms typically start at low biomass and low growth rate, increase their biomass until they've consumed all their resources. In doing so, they typically achieve a maximum growth rate somewhere along the way, as shown in the gray region. That eventually returns to 0 or close to 0. This growth rate trajectory can be mapped in a growth cone as shown below. Dynamic models typically are able to capture this trajectory, while classical FBA can provide a solution somewhere in the optimal region, as shown in the red. But it can be anywhere in that solution space. Classical FBA is not easily able to constrain it. That's where cooperative trade-off FBA or CT-FBA comes in. It introduces a biologically meaningful constraint by minimizing the distance between the point of 0 growth and 0 growth rate and the theoretical maximum as shown on the right. This approximates the trajectory expected empirically. Additionally, this constraint is, or some additional information here is that this constraint is achieved using an L2 style regularization, if you're familiar with that, that is essentially minimizing the sum of squared growth rates of the community. Additionally, CTFBA allows for suboptimal community biomass. So it could be some fraction of it, like say 70 or 80%, which helps balance the trade-off between individual growth rates with that of the community. That parameter that I was describing just a moment ago in the notebook is actually the trade-off parameter. And in our particular case, we're allowing the community to achieve 80% of the theoretical maximum, which was that 0.8 that was used in the function. Great. So cooperative trade-off FBA allows us to treat metagenome scale metabolic models with the same methods as genome scale metabolic models. So first you use CTFBA to estimate the growth rates of the community and the individual taxa. Then you can apply methods like PFBA or some minimal media style optimization to get at the fluxes. But does it work? You know, the age-old question. Here we can see some results from the original MyCom publication validating that CTFBA, the CTFBA approach that I just described. On the left, we can see distributions of individual taxa across a large number of communities and conditions. What we see is that if we don't integrate a regularization step, that L2 parameter minimization that I just mentioned, which is on the bottom here, no L2, there's a very small number of taxa that can achieve high growth rates, and a majority effectively not growing at all. Obviously, this is not what's happening in the microbiome because we observed the taxa thus, and they're not going away if you measure longitudinally, so they must be growing. Thus, the solution provided by the no L2 situation, which is essentially classical FBA, is not relevant to the system. Now, if we do apply this regularization strategy, but we still require the community to achieve its theoretical maximum, we see that there are more taxa that are able to grow. That is, the regularization better distributes growth rates across the individual community members. However, as previously noted, this maximal growth rate might not be achievable with all taxa growing. Lowering the trade-off, as you can see, even just a little, to 0.7 or 0.8, we see that almost all the taxa are able to grow, echoing that what we expect to see in the microbial community. This result by itself satisfies our assumption the taxa present in the sample should be able to grow. The team that developed this method further validated these predictions of growth rate by comparing them with replication rate, which is shown in the next two panels. For these data, the team had metagenomes, and it's been shown that coverage profiles, which is depicted in the middle here, that these coverage profiles can be a good proxy for growth rate. Using this method, the team could essentially estimate the growth rates, or in this case, the replication rates, which are a proxy for growth rates in vitro, or sorry, in vivo, since these were samples collected from a large number of individuals in a population, and we're able to compare these empirical estimates with those predicted by MyCom. And what we see on the far right in the box plot is that, again, without regularization, the correlation between the empirically estimated growth rate and the MyCom prediction is actually quite poor. If you add the regularization, but then again, require it to achieve 90 to 100% of the theoretical maximum, the correlation is still quite poor. It's when you actually allow the community biomass to, say only reach 70 or 80% of its theoretical maximum, where you start to get results where the correlation is actually pretty good. Thus, this result shows that CTFBA can provide a biologically meaningful solution to the community growth rate problem. So, in short, cooperative trade-off with L2 regularization helps us reach a solution wherein most taxa are able to grow, and the growth rates that we can achieve from this are much closer to what you might expect biologically. Easy peasy, right? So what's taking so long? Yeah, well, these methods are great and the math checks all checks out. So yeah, why is it taking so long? Well, typically Microbiome models are a bit more complex than the samples we've been using to illustrate these methods. Like, for instance, our one organism community. This visualization is one of a standard metagenome scale metabolic model. In this particular case, each pink dot represents a metabolite and each blue dot a reaction, with black lines connecting them. Each of these regions is a single taxon in the community. So you can see in the community, the reactions and the reaction network is more or less grouping into individual species or a genus. And you can see that the community model contains nearly 70,000 reactions, over 45,000 metabolites, and nearly 300,000 connections. So you can imagine that finding the optimal flux solution in this model definitely takes some computational power and sometimes a little patience. Now, before we look at our results, I know you must be excited to see what it's, how everything's looking, but and your wait's almost over. But first, let's take a look at some of the visualizations we'll create with the output from our results. Since Microbiome gives us so much great data, it can be a little overwhelming at first, but don't worry. It's actually very easy to make visualizations with Microbiome and extract the data to use with your favorite visualization package. So first and foremost, we'll take a look at growth rates of the individual taxa in each of our models. Microbiome has built-in functions for this, so it'll be pretty easy to build this style of visualization. Here we have a scatter plot with log-scaled growth rate on the y-axis for each taxa on the x-axis. This plot is a great way to check that your models are growing correctly at a typical growth rate, like what we might see here. And also tell you which microbes are doing well and have high growth rates in the particular context that you're simulating. In this particular example, we can see that Prevotella has a pretty high growth rate and seems to be thriving. We'll get an opportunity to look at this result for our communities and see how they're looking. Additionally, we'll be able to create visualizations of the niche space. This is the context-dependent sort of environment that the microbial taxon is occupying. So it's essentially how this context-dependent environment or niche space is the way in which the individual taxa are leveraging the metabolites available to it. This is a distillation of a fairly complex set of information, since plotting all the metabolite exchanges wouldn't be possible. We're using dimensionality reduction techniques to construct this plot, sort of similar to PCA. In this particular example, I'm showing you a UMAP for individual genera across a large number of samples, around several thousand. And what the UMAP is doing is it's taking those really high dimensionality data of thousands of patients with thousands of genera that each have thousands of reactions, and it's compressing it down into this two-dimensional space, sort of similar to PCA, but unlike PCA, which is assuming like a linear relationship between the data, the UMAP or TSNE, which is the method you'll see when we jump over to the notebook. These two try to preserve the distances between samples as represented by their flux vectors, but is better at capturing non-linear relationships than PCA. And so what we see in this plot is that more or less the individual clusters that are formed are representing individual genera across the many individuals, thus suggesting that the individual genera can occupy one or a few niche spaces and sort of have fairly consistent metabolic strategies that they're leveraging across populations. In this particular example, you see actually we're highlighting a few genera of interest: Bladia, Faecalibacterium, Eubacterium, and Clostridioides. And one thing that's really interesting here with regard to Clostridioides is that we see in the red that Clostridioides is able to occupy a few different niche spaces. This is true for the Eubacterium as well, and for a couple of the others. What's interesting about this is it would suggest that the Clostridioides in this case has a few different metabolic strategies that it might leverage, or different niches that it can occupy. We'll take a look at this in the context of our samples and see if we can pull out any interesting observations. Finally, we'll be able to take a closer look at the metabolic imports, which was what the previous image was showing you, but kind of distilled on a larger scale. We can look at those for individual taxa, which is what's depicted here in this heat map. We'll be able to construct our own heat maps illustrating what different metabolites are being consumed or potentially exported if that's of interest in our particular samples. In the example I'm showing you here, each row is a metabolite. Each column represents a sample with the cells colored on a logarithmic color map for the import fluxes. This actually is a representation of the fluxes for C. diff specifically, again, across a large number of samples. This is actually the complete set of data from which we're using a subset. And so here, you see that the C. diff import fluxes cluster into a few groups that are associated with its growth rate. You see there's a high growth rate cluster, a very low growth rate cluster, and then a sort of like medium growth rate cluster. Again, these kind of correspond to its different strategies. The high growth rate cluster is associated with its ability to successfully engraft in individuals, primarily those who were coming in needing an FMT. Whereas the cluster where the growth rate is fairly low, you see it's not really importing much. And for the most part, a majority of these samples came from healthy donors or people who received an FMT and transitioned to a more healthy state. Then finally, we see this sort of middle cluster where it's able to grow, but not quite as well. And this was mostly associated with the transition from pre-FMT to post-FMT. And what's interesting here is if you look at the patterns in the fluxes, you see between the high growth rate and low growth rate clusters, different metabolites are consumed, thus suggesting that maybe different niches are occupied or different strategies are leveraged. Great. So now that we have a flavor for what we're going to be looking at, we can actually go back and check on our results and try to build some of our own visualizations. Great. So let's see. It looks like the growth estimate finished. That's great. If for whatever reason it didn't work for you, you can actually just go ahead and run this next cell and it'll import the results from the treasure chest. As before, we're going to want to merge the growth rates with metadata so that we can leverage the metadata for comparisons. You can see that worked out pretty good. Also, importantly, if we look at this growth rate table, we can see the abundances that were fed in, and then the associated growth rates, and then some information about the reactions and metabolites and the various taxa. And now if we scroll down a little bit further, in addition to the growth rates, we can look at the exchanges. Again, these are the metabolites that are imported or exported from the medium. we want to take a closer look at some of these exchanges, you can actually filter based on the metabolite. Here, I'm showing you trehalose. This is a fairly interesting metabolite, as studies have shown that it can be responsible for the remodeling of the gut microbiome structure and has been associated with CDI, the prevalence of CDI. Next, we can start to build some of our visualizations. Great, so that you can see doesn't actually show you anything in the notebook. So you have to go to your materials, and then this one is called growth rates.html. So I will download it. And then if I click here, you see it opens and gives you a nice little visualization of our growth rates. And so you can kind of see who's growing well, who's not. I'll zoom out a little bit so we can get a better look. Yeah, so you can see, for instance, some common genera like Bacteroides are growing pretty well in most of our samples. And if we look at Clostridoides, you see there's kind of a range of growth here. Some are growing well, some not so much. Again, this is on the log scale. Let's see if I zoom back in, go back over here. Yeah, let's take a closer look at the C. diff growth rates. So we can use the taxon ID to filter out all the samples, all the rows in our table where the taxon is Clostridoides. So now we have just the Clostridoides across our samples. And we can use this package called Seaborn to create a box plot. And yeah, what you can see here is that if we're comparing the growth rate predicted for C. diff across the disease states of our sample, what you see is that on average, the growth rate in the pre-FMT seems to be a little higher than that of the healthy individuals, as you might expect. The pre-FMT samples are sort of in a dysbiotic state, and their microbiomes are kind of wiped out. And so you would expect there to be sort of a niche space that could be occupied by C. diff. If we want to look a little closer at the niche space, we can come down here. This function is essentially creating something analogous to that UMAP that I showed you a moment ago. So if I'll refresh here to see if it shows up. Here it is. I'll download it. And yeah, once again, you see this one is using the TS&E method that I mentioned. That's, again, reducing the dimensionality of our import flux space down to a two-dimensional grid. And if you hover over these individual dots, it kind of gives you some information about the different genera. Let's see, Clostridoides is in green. So we see there's a grouping here, it looks like. So there's a pretty big group here. It looks like it's most of our samples. And then there's a few over here that are in a sort of different group. We don't necessarily know what these groups are corresponding to, but you might be able to guess that one is probably the higher growth rate samples and the other is the lower growth rate samples. And so what this is suggesting that between those two groups, there is a different set of imports that are being leveraged by C. diff. Now, if we want to look a little closer at the fluxes, one thing we can consider is what the community itself is consuming. The state of the community, as we've mentioned, can be really important for whether or not the engraftment of C. diff is successful. So to do that, we're going to look at the global exchanges for the community to see or try to understand the state that the community is in. Now, if we wanted to do that, sort of on the fly, we'd actually want to rerun our simulations in the absence of C. diff, since looking at it with C. diff sort of wouldn't be a fair comparison. That we don't really have time for that, and so we're actually just going to pull a previously generated result from the chitter test, where we ran the same simulation but with no invasion. We're going to then use this micom function consumption rates to get the import fluxes from our data. Then there's some sort of pandas magic here happening, where we're merging our metadata as you've seen previously, and then kind of restructuring the table to make it more like a square table instead of a long table. In doing so, that's going to create a matrix where we have samples versus metabolites. Not every sample is going to have every reaction, so we're going to fill in our blanks with 0 because they obviously just didn't show up. And then one thing that's very important here is that we do a normalization. The fluxes kind of vary exponentially, and so to normalize them, we're going to want to apply a logarithmic transformation. That way, they're sort of they're more normally distributed. And then finally, we will sort the fluxes by the variance, with the highest variance first. So if we run this, you see we get a table where we have the different metabolites as on the index and the samples in the column. And you can see the fluxes associated with the different metabolites across the samples. Here, negative values are small and positive values are large. So already just kind of looking at this, you see there seems to be a difference in glucose consumption across our samples, with glucose being pretty low in the pre-FMT and much higher in the healthy samples. But let's make a heat map so we get a better visualization of that. Great. So yeah, what we can see here is that in the imports of the community seems to cluster based on the disease state, with the healthy samples on the left for me and the pre-FMT on the right. Because the clustering might vary slightly with everyone, it's possible this could be flipped for other people. But you do see there's a difference, and just kind of looking at the heat map, you can see pretty clearly that things are different. Again, I mentioned glucose. That's one difference. You can see, though, there's quite a few other metabolites where the flux is either high in the healthy sample and low in the pre-FMT, or vice versa. For instance, you see acetate here seems to be pretty high in the pre-FMT, but absent in the healthy samples. Again, just suggesting that the communities are in a different state, which maybe is making them more or less susceptible. Great. So now I'm going to just briefly hop back over to our slides. And now it's your turn. So next, you're going to have the opportunity to kind of figure out how to create some visualizations yourself using Micom to investigate the niche occupied by C. diff specifically in our samples. So just briefly, I'll touch on that. So if you scroll down a little bit further into this exercises portion, you can see this challenge here, investigating the metabolic strategies for C. diff. Here I've set up sort of a skeleton for you to use. And then you see there's portions for you to kind of fill in your own code. This is going to be producing an output that's sort of analogous to this heat map, but looking specifically at the imports of C. diff. So again, you can fill in your code here for these individual steps to get it to run. And then, yeah, try to make a heat map for yourself. And so with that, I think I will go ahead and take a break. so that we can give you guys the chance to hack away, and we'll be back shortly.
Say we'll be back at 20 for final check over the exercise.
Great. Yeah, we'll be back at 11:20 for a final check-in to see how everyone's doing and see how the exercise went. All right, see you then.